So a lot has happened even in the last 3 months, especially at DDW, that really kind of casts an even greater light on the microbiome and its role in IBS, and particularly small intestinal bacterial overgrowth. So I look forward to the question and answer, which I hope I'll provide. But we're gonna take this till 6 o'clock, probably this presentation, and then if I can get the slides to move, and then we'll take question and answer. But a lot of new stuff is coming near the end, so stay tuned for that, because it's going to be very impactful. But I put a new slide in here compared to previous journal clubs that I've done, because I think it's good to go back to 1996, because in 1996, this was what was being done for IBS. This is what they thought was causing IBS. I'm not kidding. This is what one of my senior colleagues used to say, and still sometimes says, about IBS as a disease of hysterical women, which is really a terrible thing to say even in 1996, but this is what was being described. And if you look on the right side, this is what was being given: psychological therapy, antidepressants, antispasmodics, antidiarrheals, and laxatives. Recently, the AGA has put out a guidance on irritable bowel syndrome, and still antidiarrheals come up on top as a guidance for treating IBS with diarrhea. So Imodium, those sorts of things. Well, could you imagine that a Crohn's guidance from the AGA or the ACG puts Imodium as their first-line therapy because it's cheap? It wouldn't happen, no chance. Even though Imodium would slow the diarrhea of Crohn's disease, it's not going to happen. You're not treating the cause. And yet here we are in 2023, still putting Imodium first in a guidance, and that makes no sense to me whatsoever. We really need to treat the disease, not treat the symptoms. Anyways, that's my shtick. I'm gonna stick with it. Okay, so I'm gonna talk about IBS pathophysiology. This is what I'm gonna cover today. That food poisoning, particularly E. coli, Campylobacter, Shigella, Salmonella, has a particular toxin, CDTB, which I'm going to talk a lot about. And exposure to that forces you, or convinces your immune system to make an antibody to vinculin, leading to nerve damage and changes in the gut neural structure, specifically the small bowel, leading to bacterial overgrowth. We're going to talk a lot about that, and that IBS is an antibiotic responsive condition because of that. Well, let's start with the two bookends. The bookend is, does food poisoning cause IBS? I could speak for an hour on this or longer, but it really boils down to this paper by the Mayo Clinic, by Clem. Focus on the top part, which is bacterial gastroenteritis. And the bottom line is, if 100 people go to a wedding and get food poisoning, 11 of that 100 are going to end up with irritable bowel syndrome from that food poisoning. That's the risk. So it's real. Food poisoning causing IBS is real. But I want you to understand that the whole premise of IBS forever is that it's the leftovers. You have a colonoscopy, there's no Crohn's disease. You biopsy the small bowel, there's no celiac disease. Okay, well, it's IBS. Well, if you put 100 IBS patients in a basket, not all of them are going to be microbiome. Some of them are going to be different conditions. So we knew IBS isn't going to be one thing, it's going to be multiple different things. But the question is, what percent of IBS is gastroenteritis? Well, we actually modeled this, and using a very complex mathematical model, we took 307 million people, adults, plopped them into the United States, 0 have IBS, and then we overlaid the CDC, because food poisoning is a reportable illness, the incidence of food poisoning, and then modeled it over time. And what would happen is 9.1% of the entire US population could have their IBS explained by food poisoning based on our assumptions. So that's 60% of IBS at the time of this study using the Rome 2/3 criteria. So food poisoning causes IBS. That number 60% seems to creep up over and over again, as you'll see in this presentation. But can food poisoning, by causing IBS, work through the production or forcing you to develop small intestinal bacterial overgrowth in some way? So this goes back now to 2008. This is a study. We gave rats Campylobacter. We gave another set of rats placebo. They overcame the Campylobacter 1 month later, and then we waited another 3 months for a total of 4 months to see the development of what happened to these animals. Did they get IBS? Not only that, we looked for overgrowth. So 27% of the rats who got Campylobacter now, 4 months later, have bacterial overgrowth. More importantly, the rats that got Campylobacter and developed overgrowth, those rats had altered stool consistency. So essentially, they had IBS-like stools. They can't tell you, they can't fill out a questionnaire, but they had altered stool consistency and increased rectal lymphocytes in that group also, which is the only other finding in post-infectious IBS in humans. So why is this study important? This study is important because it's the first time we have an animal model using Campylobacter, the #1 cause of food poisoning in the United States, therefore the #1 cause of IBS in the United States among the food poisonings or gastroenteritis. So we can now see that it is causing IBS in this model. But this model doesn't stop here. I'll keep going. You'll see more. Fast forward to 2022, all the work done with this model and various other studies, some of which I'll touch on again later, you can then apply what's called the Bradford-Hill criteria. Bradford-Hill criteria is that you take all the data that's available and you have to meet certain criteria, which are on the left column, in order to say, okay, Campylobacter causes IBS. And the bottom line is there's evidence for every single criteria for the Bradford-Hill. So basically, Campylobacter causes IBS. It meets the Bradford-Hill criteria. And this was published in 2022. But how is this working? So how do you get from food poisoning to bacterial overgrowth and therefore IBS? Well, I told you Shigella, Salmonella, Campylobacter, E. coli, all of them can cause irritable bowel syndrome. But they all share one toxin in common called cytolethal distending toxin B. And that toxin is unique to all of them. They all have it, even though they look very different, as you can see on the screen. It turns out when you get exposed to CDTB, a part of that toxin looks like a human protein called vinculin. And vinculin is, if you look at these are cells in our lab, the green is the skeleton of the cell or actin filaments. And at the end of the actin, there's a little red tuft, or you can see it better here, or you can barely see it, but it's there. And the tuft is vinculin, and it acts like a motor and moves the cells to attach to the next cell. And the particular vinculin for which antibodies bind to vinculin is a 117 kilodalton vinculin present in the nerves of the gut. So that's really important, and you'll see more information about why that's important. But what happens to the nerves of the gut, going back to our Campylobacter animal experiment, is that these nerve cells, the deep muscular plexus, DMP, interstitial cells of Cajal, are the nerve cells of the gut that make the small bowel cleaning wave work. So the small bowel cleaning wave is necessary to clean the bowel clean the small bowel between meals so it's ready for the next meal. This is controlled. You can see there's a lot of cells all lined up and attached to each other. Here is Campylobacter treated, but they didn't get overgrowth. And these rats were the ones that got overgrowth after Campylobacter. Maybe one cell here, maybe barely one cell here, but they're far apart. They're not connected. And you can see the cells, the density of the cells lowest in the group that got the Campylobacter and developed SIBO. What does that mean? So those cells make the cleaning waves go, and they're reduced in the animal model. Well, we already knew from 2002 that in IBS with SIBO, these are humans, human IBS with SIBO, the number of cleaning waves, migrating motor complexes of the small intestine are dramatically reduced. Not just dramatically reduced, half of the IBS patients have no cleaning waves detectable at all in this study. So no cleaning waves equals SIBO. That's been known since 1977. this interdigestive motor complex, migrating motor complex, housekeeper wave, cleaning wave, whatever you want to call it, it's all the same thing. When it's not working, you get bacterial overgrowth in the small bowel. Okay, so let's put this a little bit all together. I've told you the CDTB is important. I've told you Campylobacter can cause IBS, but is it Campylobacter or is it the CDTB toxin that's doing it? Well, this is the study. So we took the CDTB toxin only, and we injected it into this under the skin of rats, excuse me, and 3 weeks later, we injected a booster. Just like the COVID vaccine, 3 weeks later, you get the booster. And what happened to these rats? Well, they developed antibodies to CDTB, of course, because we put CDTB. This is the antibodies before in their blood, and now their antibodies are way up here. They then developed antibodies to vinculin, so they developed autoimmunity to vinculin. We didn't give them vinculin, we just gave them CDTB toxin, so they developed antibodies to vinculin. And they developed overgrowth. So I want you to want to say it again so you understand. We put a toxin from food poisoning under the skin of the rat, not in the gut, but the gut became dysfunctional from the antibodies produced, leading to overgrowth of bacteria in the duodenum and the ileum, and a reduction in vinculin in the small bowel tissue. So it's the antibodies to CDTB that were causing this change to the gut physiology. So it works like this. You get exposed to the CDTB toxin. The antibodies don't like any of the toxin because it's a foreign agent to you. But one of those areas looks a little bit like vinculin. Supposed to go over there. There we go. Looks a little bit like vinculin, and so you develop autoimmunity. And that's well known in the autoimmunity world as molecular mimicry. The toxin is mimicking you to get you to form autoantibodies. So now I'm going to take it to another level. And this is a repeat of that trial, bigger, where we're again immunizing rats to CDTB. This is really important for you to understand because this animal model is so much like what's we're seeing in humans that is really remarkable. So here we're giving the CDTB again. So you're going to see it again. Control rats, no antibodies to CDTB. The CDTB retreated rats, higher antibodies. The control rats, not having diarrhea. The CDTB treated rats now, sometime later, having wet stool. It's statistically more watery stool. And then the antibody level predicts the higher stool wet weight. So the higher your antibody, the more your stool wet weight is in these rats. Then we looked at the microbiome, which we hadn't done in the previous study. So this is deep sequencing of the microbiome. You're not changing the microbiome of stool or the cecum even. You're changing the microbiome of the small bowel. That's what's happening with this toxin. It's affecting the small intestine. But here is exactly what's happening to the microbiome in these rat studies. You have control rats. They didn't get anything. This is where they're sitting in green dots. Then you have a group of rats who got the toxin and they didn't change. So they're the people at the wedding who got Campylobacter, but oh, I'm fine, I didn't get IBS. But the ones that did get IBS went into two directions. They went either high E. coli here, and we know E. coli is a hydrogen producer, or high hydrogen sulfide production, or desulfovibrio. So the rats were developing abnormalities in their microbiome in two microtypes, hydrogen production or hydrogen sulfide production, because of the types of organisms that were overgrowing. Okay, so remember that because we just had a microtype paper in human stool that exactly shows this. So it's the same. Okay, I'm showing you this. This is a poster from DDW just a few days ago. I'm not going to get into the details of this, except to say, if you give CDTB, when we look at the tissue, again, we're injecting the CDTB into the arm of the rat. And if you look at the tissue of the small bowel, you're getting changes in pathways related to, as you can see down here, related to circadian rhythm, related to barrier function. So you're losing the barrier, which is sort of a leaky gut kind of concept. You're changing the motility, so chemicals and factors that are important for GI motility are affected, and chemicals that are associated with visceral hypersensitivity are affected. So CDTB is causing all the things we know to occur in humans with IBS. Okay, so now let's go back. Can we measure these antibodies? Can we determine that you have IBS measuring CDTB and vinculin? But I want you to rethink about this. I'm not looking for IBS. I'm looking for post-infectious IBS, right? So that's about 60% of the 100 IBS patients. So bear that in mind as we go through this data. So we developed a first generation test, which I'll talk about in a second, but it wasn't as good as this. We had to improve the test because the proteins are funny, and we had to do what's called epitope optimization so that your blood, your serum, would see the toxin or the vinculin and be able to bind to it in an assay on a plate. And so with epitope optimization, we got head and shoulders differentiation of IBS in red versus IBD in all the other colors. But it boils down to this. So this is what I want to show you. Medical certainty is 80% or above. So whether you're CDTB positive, vinculin antibody positive, or both, your post-test probability of IBS is medically certain. So that's really important. But remember, if you look at sensitivity and specificity, and if you look at the pooled either or, it's 56%. So 56% sensitivity sounds low, but I said 60% is the number of IBS patients with food poisoning as a cause, we believe. So 56% is really close to 60, and that's the exact number we would want. So don't focus here, focus on specificity and post-test probability. But here I want to show you the first iteration of the blood test for IBS. The old assay, I call it, the generation 1. This is the likelihood ratio. Likelihood ratio is what determines post-test probability. For CDTB, it was 5.2. With the improved test, 6.3. With vinculin, it was 2. With the improved test, it's 5.3. So we've improved it a lot. And with this epitope optimization. So really, the only test for anti-CDTB and vinculin that you can use these numbers on the screen are the second generation test. And I'll show you what it means here in just a moment. So let's look at post-test probability of any test ever available for IBS. There was a pain perception test. 55% is pre-test probability of IBS based on a clinician. Pain perception gives you nothing. There was a 34 biomarker panel, gets you to 74% post-test probability. Bile acids in stool gets you to 78, still not medical certainty. 10 biomarker panel gets you to 79.4. Visceral hyperalgesia, when you put a balloon in the rectum, which nobody does, you can get to 82% if you detect visceral hyperalgesia. Volatiles in the stool, not bad, getting to 86%. Not available, but it's not bad. The first generation blood tests for CDTB and vinculin are not making the mark. 60 here, which is almost as good as nothing, and then the CDTB here. But the second generation test, hands down, is the best. It's the best test on the market for irritable bowel syndrome with diarrhea. That's what it's used for, and it's detecting post-infectious IBS. So this improvement in the test really has made a dramatic difference. Okay, so it works like this. You're fine. You have this beautiful microbiome. Then you get Campylobacter. You're exposed to this toxins, the CDTB toxin. You start to form antibody to it very quickly as you're infected and recovering from a Campylobacter infection. And then later you're forming the antivinculin antibodies, and you start to lose the nerves of the gut. Specifically those nerves that are responsible for the cleaning waves. The gut doesn't function right, the cleaning waves don't occur, and you get a buildup of bacteria called bacterial overgrowth. This was published in Pathogens and Disease. So that's the story on antibody testing for IBS, and that's the story of the IBS SMART test. Well, let's talk about breath testing. So I was at DDW, and I was shocked to see a speaker at DDW say, well, we don't know if breath testing is more increased in IBS versus healthy controls. And I'm like, well, if you look on PubMed, which is a website, you can see this paper, which just got published in 2020, and you can see clearly that the meta-analysis from this Australian group is breath testing is more abnormal in IBS versus healthy controls, period. And so we now know this is true, and likely, and I'll show you what the culture data shows, we're hitting the same 60%. So I'll get to that in a minute. The problem with breath testing is back in the early days, all we had was hydrogen. And that was wrong, because hydrogen doesn't do as much as the other two gases. And the other two gases use hydrogen to make methane or to make hydrogen sulfide. So the more methanogens you have, the less your hydrogen is going to be. So if you don't know the methanogens, you can't trust the hydrogen numbers. If you don't know the hydrogen sulfide producers, you can't trust the hydrogen numbers, because it's going that way. Hydrogen is not preserved, it's being used. We now know hydrogen sulfide is associated with diarrhea, and we know methane is associated with constipation. That's clear now in 2023. Well, let's go through the small bowel culture studies. So people say, well, culture is the gold standard. Okay, so let's look at the culture studies. Higher in the small bowel of IBS versus controls in this study. This is the most important culture study in the sense that it gets us our number of 60%. So these were DIBS patients versus sick people coming in for endoscopy. And you can see 60% meet the threshold of greater than 10 to the 3 bacteria per ml in the small bowel, which is the new standard for SIBO. Now let's get to the Reimagine study. So at Cedars, we've been doing this Reimagine study now, I want to say 5-6 years, maybe longer. We're trying to get as many small bowel aspirates as we can from patients coming just for upper endoscopy. And these are the diseases we're very interested in the context of small bowel microbiome. But focusing on SIBO, when you do 16S sequencing, I'm going to explain 16S sequencing, and I'm going to explain shotgun sequencing because they're different. 16S sequences the ribosomal RNA of bacteria and is able to fairly well categorize the bacteria based on those sequences. So this is 16S 2020. SIBO on the right by culture, greater than 10 to the 3. Non-SIBO on the left. And every ring I put, so the middle ring is the kingdom bacteria, the next ring is phylum, etc., 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 until you get to genus and species. And you can see there's a lot of blanks as you get further outside on the ring, because 16S sequencing can't get you the depth to be able to determine the genus and species in all instances. But what we knew back then in 2020 is that this was Klebsiella and this was Escherichia. That's what we knew back in 2020. But what we were able to do with that same study is say, okay, if you do a lactulose breath test, not a glucose breath test, a lactulose breath test, at 90 minutes, if you're more than 20, which has been the criteria for this test, you get the best test dynamics, the best correlation with the sequencing. But look here in the last two columns. You're able to look at the metabolomics of the bacteria in the small bowel, and that correlated with hydrogen producing functions in the small intestine being elevated. So the hydrogen on the lactulose breath test that you see at 90 minutes is correlating with what's going on in the small bowel, not the colon. So people who argue, well, it's a measure of transit and all this stuff, it's not true. It's coming from the small bowel. And we have more data saying it's not coming from the colon, but I'm not going to get into that. So DDW, just a few days ago, we were able to show for the first time, using both sequencing and culture of the small bowel, exactly how to do culture properly, because nobody knew how to do it properly. Because there are some centers that are doing culture. They don't do breath tests, they culture. And they combine blood agar and McConkey agar, and they add it all up. And how can you add blood agar and McConkey agar when blood agar picks up the same bacteria as McConkey agar? You're double counting. Doesn't matter. We settled the issue. Blood agar doesn't mean anything because it doesn't correlate with the higher the bacteria, a change in the microbiome. McConkey does. So the higher your bacterial count on McConkey agar, the more deteriorated your microbiome. And I'll show you what's going on in more detail on this slide. On the left side, this is the microbiome of a normal human. In the middle is the microbiome of somebody with more than 10 to the 3, but less than 10 to the 5. The lines that connect the dots are bacteria, the lines are the connectivity of the network. So think of this like a city. You got doctors, you got plumbers, you got sanitation workers, you got nurses, you got whatever, bakers, etc. It has to be very tightly interconnected and very broad. And so the more dense the connections, the nicer the network is. But immediately at 10 to the 3, the network starts to fall apart. Greater than 10 to the 5, it's fallen apart completely. So the higher the bacteria are on McConkey agar, the more the microbiome has been destroyed or disrupted in the small bowel. I'm going to come back to that with even more data on shotgun sequencing. We're going to shift gears to intestinal methanogen overgrowth. This is important. We changed the name from SIBO with methane to IMO, intestinal methane or methanogen overgrowth, because the methanogens aren't in the small bowel like that. They're everywhere, or at least we suspected they were everywhere. But we know this is the character. This is the prime suspect producing methane in the gut, called Methanobrevibacter smithii. It's not a bacteria, it's an archaea. It needs hydrogen to make methane. When it makes methane, it produces constipation. The higher the methane, the higher the constipation. We've shown all this work previously, and I'm not going to get into all that because this is methane in and of itself is 1 hour of talking. But what we did show at DDW for the first time is we did sequencing of the duodenum and sequencing of stool to see where these methanogens live. And Methanobacteriaceae, which is M. smithii from the last slide, is present in the duodenum and stool. So when it's up in all segments of the gut is the point. And there are more details on this slide that are interesting, but I don't want to bog you down with too much details here. The third category is hydrogen sulfide, and one of the key organisms is Fusobacterium varium, and it eats hydrogen or consumes hydrogen to produce hydrogen sulfide, and the outcome of all that hydrogen sulfide is diarrhea. So let's talk a little bit about hydrogen sulfide before we start getting into the technology. Well, how to develop the technology is really what I want to get at here. So we had hydrogen only in the 1980s and early 90s. Then we had hydrogen and methane. And now we have hydrogen sulfide, hydrogen, and methane. But this was not easy. These are easy. Hydrogen sulfide is not easy because it's a reactive gas. It converts into other things very easily. So you didn't have a prior technology to transport hydrogen sulfide that worked. We checked. Hydrogen sulfide is very reactive. You need a special material to harbor hydrogen sulfide without changing the concentration in the gas. Then you need to array 4 sensors in the instrument, a new instrument that measures all 4 gases, the 4th gas being carbon dioxide, but measuring the 3 important gases, hydrogen, methane, and hydrogen sulfide. And they have to be arrayed in such a way so they don't cross-react with the other gases. And then you have to do research to show that using the bag, mailing it in, stability, that the numbers that you get correlate with symptoms. And all of that had to be done, and that took about 5 years. So now we initially had the cutoff at 5 parts per million till we had more science. Then it was 3, and I'm going to show you some evidence for 2 here in a moment. This was the first study taking diarrhea patients, not Crohn's, not ulcerative colitis, but functional diarrhea patients, and they're high, over 5, compared to the constipation, functional constipation, and healthy people. But I'm going to show you 2 in a minute, so give me some time. This is another study showing the higher the hydrogen sulfide, the more severe your diarrhea is. But this is the study I want to show you, and this is really important. We took patients from a IBS study. We didn't select them for microbiome. They were just here for a double-blind study on diarrhea IBS. We took patients from another study, a double-blind study on constipation IBS. Again, we didn't select for microbiome. It's just a constipation IBS study. And we did the 3-gas breath test on these patients at baseline. And so I want to show you those results, which was published in 2022, and the microbiome, because we have microbiome on these patients. So here is the data. So if you're a diarrhea patient, which is the blue line, that's what your methane looks like. Not there, no methane. Among the constipated patients, not all of them had methane. About 60%, between 50 and 60% had methane. And this is what the methane looks like when it's positive, okay? Look at hydrogen. Hydrogen, by 90 minutes, the average IBS-D patient is more than 20, which is the cutoff for SIBO. Look at the patients with methane. The hydrogen is low, statistically low, even lower than the non-methane constipators, because methane eats hydrogen. If all you measured with methane, you wouldn't know what's going on. If all you measured with hydrogen, you wouldn't know what's going on. So you have to have both. Same thing for hydrogen sulfide. In diarrhea, hydrogen sulfide's higher than in constipation or the other category. So hydrogen sulfide and hydrogen are present in diarrhea. Methane is present in constipation. But here we go one step further. In the methane positive group, M. smithii in the gut is higher. Not only is it higher, that the methane correlates with the methanogen in the gut. There's a direct correlation. Really important. Here's the next part of it. We were able to look at the specific methanogens, and they correlate with methane on the breath. And then we were able to find specific bacteria that feed hydrogen to methane, and we don't need to get into that, but that's an interesting part of this. On the hydrogen side, the hydrogen negatively correlates with methane because methane's eating hydrogen. Fusobacterium correlated with hydrogen sulfide. And the pathways in the gut are upregulated for methane and methane synthesis when methane's positive on the breath test, and hydrogen sulfide correlates with hydrogen sulfide producing pathways being elevated in the gut. So I'm saying a lot of words, but this is the first study. So basically, you fill a bag with your breath, you send it to a lab, you run it on a 3-gas instrument, and that system, for the first time in the history of breath testing, correlates with what exactly we see in the microbiome. So this 3-gas breath test, the way it's designed and the way it works, correlates exactly with your microbiome. We've never shown that before. So it really validates breath testing. Breath testing is representing what's going on in the gut in this gas test, this 3-gas test. So no other breath test has been validated to this degree that this system works and shows what's really going on in the microbiome in this way. So a real challenge to do all this work, but when you do the science, you prove that this system, this bag, this way to do it really works. And you end up with 3 microtypes. Remember I told you about the animals? They get a hydrogen microtype and a hydrogen sulfur microtype from post-infectious IBS. Same thing in humans. Hydrogen microtype, hydrogen sulfide microtype. Food poisoning doesn't cause methane, but the 3rd microtype is methane, which causes constipation. One last thing at DDW, we took AI, used AI, an app called Dieta, where the patients at home take pictures of their stool. And the app is able to calculate a number of parameters of the stool. So another validation of this system of 3-gas breath testing is that the methane correlated with the change in Bristol stool score, meaning a lower Bristol stool score, and less edge fuzziness, which means the stool is like so tight you get no fuzziness on the edges of the stool, and less bits and pieces of soft stool. Here's an important part, is that the start of higher volume stool is at 2 parts per million, not 3 parts per million. So as soon as you get above 2 parts per million, the volume of stool goes up. So now in my practice, I'm looking at 2 parts per million as the determinant of an increasing diarrhea-like stool volume. So we were able to learn this for the first time and presented this at DDW. But hydrogen sulfide is associated with diarrhea even compared to hydrogen-positive patients. Adding that hydrogen sulfide makes the diarrhea worse. So again, never in the history of breath testing were we able to show that a specific test was able to correlate so well with the microbiome and the human condition. I'm not done. I'm not done. Sorry. So this is shotgun sequencing now. Shotgun sequencing is not 16S, not RNA of the bacteria, but the actual genome of the bacteria. And by doing this, we're able to determine exactly what bacteria are there. I'm going to skip this slide and go to the next slide and see now we can add an additional ring to this circle, which we couldn't before, to get to not just species, but even the strain of bacteria. So what did we find in SIBO? This is non-SIBO, this is SIBO. Again, proteobacteria. Look, proteobacteria, let me just click on this because you got to see this. More than 50% of the entire microbiome in a SIBO patient is proteobacteria. I mean, we didn't even imagine this was going to be the case. Like, we were thinking, okay, it's going to be a lot of proteobacteria, but this shocked us that it's so dramatic what happens when SIBO happens. Let's dissect this more. So SIBO, now we understand, is Klebsiella and E. coli, but when you put those two together, you get 50%. But look at the specific strains and species when you add the additional information. 18% of everything in your small bowel now is Klebsiella pneumonia, one species. The other species that takes up the other percentage is E. coli. 28% of every single organism of 500 different organisms in the small bowel now is E. coli. It almost feels like an infection, right? Because this is pretty dramatic. So in the entire reimagined cohort of 800 patients, nobody's like this except SIBO patients. Only SIBO patients have this destroyed microbiome with E. coli and Klebsiella. It's absolutely clear who has SIBO and who doesn't. And you can see all the different strains we're able to identify. When we compare those strains to symptoms, there's a clear correlation, especially E. coli K12, bloating, excessive gas, abdominal pain, diarrhea, urgency. Klebsiella aerogenes, gas and abdominal pain and diarrhea are all correlated. But then when you look at the metabolome of the microbiome, this is super interesting and was surprising to us as well. Not only are Klebsiella pneumoniae and Escherichia E. coli elevated, they're jacked up to digest carbohydrates. If you had your beautiful regular microbiome and it wasn't high in number with these two organisms, you digest carbohydrates, but at a slow pace. But when these two guys come in, they can fire through carbohydrates so fast because their systems are augmented for carbohydrate digestion. 63 times higher ability to digest carbohydrates like glucose. So it's not just that you get bloated from sugar. You can super ferment, which is really amazing for us to see. And hydrogen sulfide production is augmented in this microbiome as well, and other things which are too much for this talk and will be interesting in the coming months and years. So let's go back to our network and let's just add E. coli. And when we look at our Reimagine study, when we add increasing E. coli only, the network breaks down and breaks down further. If we add Klebsiella, it's even worse. Klebsiella, even a little bit of Klebsiella, destroys the microbiome. So we call them disruptors. They're taking a beautiful garden, and like weeds, they're just destroying the garden. The vegetation does not grow. They just take over. So now let's take it easy on these last few slides and sort of say, okay, so now let's talk about the consensus documents. We have the 2018 consensus. Now the Europeans have a consensus on breath testing, and the Asian community, including Australia, have a consensus, and they are a consensus among the three of them because they all mostly agree. This is how you're supposed to diagnose SIBO. They agree on that process. They agree on the substrates, and they mostly agree that SIBO and IBS are now, there's enough evidence that they are interrelated. So here we are, three gases, hydrogen, hydrogen sulfide, and methane. You have to measure all three, otherwise you're missing things. This 3-gas breath test is the only breath test in history that's validated against the microbiome and the clinical symptoms all at once. This is the sequence, IBS pathophysiology, from food poisoning to the development of IBS through bacterial overgrowth through the CDTB toxin and antivinculin antibody. How do you treat this? Well, IBS, rifaximin, this is rifaximin study. Rifaximin is FDA approved for IBS on the basis that IBS is a microbiome condition. But we now know that if you have IBS in the Target 3 trial and you don't care, 44% respond. But if you do care and you do a breath test, 56% of those patients respond, and 76% responded to rifaximin if rifaximin made the breath test normal. So knowing the breath test augments your response to therapy. For methane, we have this double-blind study where here's neomycin alone versus neomycin plus rifaximin, and that made your constipation better in methane-positive people. For hydrogen sulfide, something's coming. Stay tuned, and we've already done some studies in it, but what we do now in clinic, I use it based on this study from Michael Levitt, some of you may know him, using bismuth with rifaximin, and I'll show you how I do that in a minute. So this is what I do in clinic based on all the evidence that I've tried to present to you today. If you have chronic diarrhea or mixed, everybody gets an anti-CDTB antivinculin because I want to know why they have IBS. I want to settle the patient into the diagnosis. I want the patient to know that if you get food poisoning again, these antibodies are going to go higher and it will make things worse. So I counsel them in travel and eating behaviors so that they don't get food poisoning, or at least they reduce their risks. I do the 3-gas breath test. If they're hydrogen positive, they get rifaximin. If they're hydrogen sulfide positive, they get rifaximin and bismuth in my clinic. And I have a number of accumulating patients who have H2S, who've gotten the rifaximin before, it didn't work, but rifaximin and bismuth, the diarrhea is gone. So I'm starting to accumulate those patients to have a case series so we can have a publication very shortly. Constipation side, I don't do the blood test because it's not present that often, and food poisoning does not really lead to constipation. It leads more to this top part. But I do the 3-gas breath test because there are some patients who have all 3 gases. When you have all 3, methane always wins, meaning you have methane, you're constipated, but you can't go from constipation to diarrhea. So it's better to know that you have all 3 gases and try to deal with that. But if you have methane positive, it's rifaximin plus neomycin. Methane negative, think of something else that's causing the constipation. Maybe they have an anal rectal disorder. So I started with this slide on the diagnosis and treatment and the pathophysiology of IBS circa 1996. I'm not going to talk about it again, but this is where we are today with the microbiome. A lot has happened. We understand this process very deeply. There's more to learn always, but I'm very excited about the future of IBS. So in conclusion, IBS is a small bowel microbiome disease. SIBO is an important contributor to that and to IBS. The most important organisms we now know are E. coli and Klebsiella pneumoniae in the small bowel. They produce hydrogen. And if you reduce that hydrogen, the IBS gets better. Methane, and we now know the bug, Methanobrevibacter smithii, is associated with constipation. More methane, more constipation. Reducing methane reduces constipation. Hydrogen sulfide is key to understanding it more completely, especially the diarrhea side. In our clinic, when we reduce hydrogen sulfide, the diarrhea improves. The new 3-gas breath test is the first and only breath test validated against microbiome and symptoms simultaneously, and hydrogen sulfide is really important. You have to know all 3 gases. The second generation anti-CDTV and antivinculin tests are the most accurate tests for IBS thus far, and it's really looking for post-infectious IBS. So now that we have all this information, new treatments are already emerging and on the way. You'll hear more about them in the coming year based on the pathophysiology that I've showed you today.